the design of the field step in metabarcoding studies is of great importance, as it can directly affect the statistical analysis power and analytical interpretation of all subsequent steps. For example, the sampling size and the number of biological repeats are directly proportional to the probability of detecting the target taxon. The technique is currently used by, sampling technique is currently used by precipitation of DNA from small volumes of water samples, like 10 or 20 milliliters, or by filtering the water samples or soil samples or air samples through filters of different sizes from 0.22 to larger pore sizes. The eDNA exists in environment in two different forms, as we discussed. It's inside or outside the cell. In the extraction of eDNA in these two different forms, if DNA is inside the cell, like within the mitochondria, for example, are desired to be obtained extraction from the filter. If extracellular DNA is the target, then it's recommended to apply centrifuge-based, settling-based, or isolation protocols. In the results obtained from the studies in the literature that are published, it was determined that a small part of the eDNA obtained from fresh water was found in genomic form. And it was revealed that this situation was of cell and organelle origin with preserved integrity. Thus, eDNA can be found in environment in both degraded and intact forms. However, the results of many research on the origin, condition, and the way of obtaining eDNA have largely guided many studies, like filtering, replication numbers, sample volumes, and spatial sampling strategies. Although the eDNA method has many advantages, it also has various disadvantages, such as obtaining false negative and false positive results. The most important step of keeping these disadvantages at the lowest level is the correct sampling, the designing your sampling strategy very well. The amount of the eDNA obtained from macroorganisms is usually obtained at very low concentrations, like lower than 200 picograms per liter. And these eDNAs are heterogeneously distributed throughout the environment. Therefore, the species identification in studies conducted with eDNA sampling depends on the ability to obtain eDNA. This possibility depends on not only on the presence and the concentration of eDNA in the sample, but also on the filtration technique, isolation efficiency, sampling protocol, and the specificity of the study. In order to increase the probability of detecting the target species, the possibility of being affected by the characteristics of the area, such as the physicochemical parameters of the water, should be taken into consideration. And the sampling and analysis protocols should be optimized by taking these into account. Preliminary work after the design of eDNA studies is recommended by all researchers working in this field. Due to high sensitivity of the eDNA methods, establishing a sample collection protocol from clean and consistent sampling site minimizes the possibility of contamination. These protocols should include negative site controls and report details of actions taken in the studies. In order to avoid any contamination between sampling and laboratory studies and to maximize the DNA copy number, the field equipment or consumables and work areas, they should be all separated from each other with well-optimized protocols. disposal of equipment and consumables used to decontaminate, including the boots you wear in the field and other gears, is of paramount importance to maintain the sample independence. Typical field decontamination protocols like 10% bleach solution or ammonia may be insufficient to meet these requirements. Disposable materials for eDNA studies can significantly reduce the risk of contamination. However, if some consumables like water sampling bottles needed to be used more than once, the items should be cleaned with a 50% bleach solution and truly rinsed before reuse. Negative field controls are critical to detect the contamination. Clean water collected using the same protocol and equipment should be preserved and treated as in the same samples. Taking separate negative control samples at each stage helps identify the source of contamination when any contamination is detected. Alternatively, taking negative control samples in the first step and subjecting them to the same protocol as samples across the whole field provides a comprehensive negative control at low cost. But of course, it provides less information about where the potential contamination has occurred. In conventional PCR analysis, when a positive result is obtained for a species from any of the negative control samples, all associated samples that yield a positive result for that species should be viewed as a suspect and should be repeated. The number of negative controls required at each step should be determined according to the number of samples and the establishment of the confidence interval. As we know that the eDNA begins to degrade immediately after being released into the environment, this degradation continues after sampling due to reasons such as mechanical forces, microbial activity, the nucleases, and the chemical reactions like oxygenation. Therefore, samples should be preserved as soon as possible after collection using a standardized protocol. For example, 24 hours on ice until filtered is a very common protocol for the time to store the samples until you reach the lab. Some protocols state that the samples should be filtered within 16 hours. Precipitation and filtration are the most common methods used to extract eDNA from environmental samples. And precipitation involves preserving a small amount of sample by adding a salt-containing solution, typically it's sodium acetate, and pure ethanol, and kept them at -20 Celsius degrees. Another protocol is to centrifuge the water samples, which were stored in ice, shortly after the collection, and then preserve the resulting pellets with 95% ethanol or another DNA preservative like DNA guard or fixed DNA or fixed RNA or RNA later, the ones that I remember now. So the filtration method is to filter and store the environmental samples obtained in larger volumes, like 250 milliliters to 2 liters, but depending on the water type and your filter, it can be up to 100 liters in marine environments, for example. The biggest advantage of the filtration method in the field is that it can be taken into the protective solution as soon as possible after the filtration of obtained samples. So alternatively, the filtration method in the laboratory can save time as it allows samples to be filtered at the same time in the field. At the same time, after filtration in the laboratory, the filters can be stored directly in the freezer, ethanol, or in a lysis solution like Longmire's. Examples of eDNA are eDNA filters are cellulose nitrate, fiberglass, polycarbonate, nylon, and polyether sulfone, and cellulose. Since the most free DNA molecules are lost during the filtration process, the pore size of the filters are of great importance in the design of the studies. In some cases, filtration can enable more DNA to be obtained from the precipitation water sample. Pore size is not sufficient to obtain more DNA. Isolation methods, extraction method also affects the final amount of the DNA. Sampling and conservation methods are compared to find the maximum efficiency that might differ by system and target species. Suspended particulate matter can clog I think there is a disconnection. I was talking about sampling and conservation methods are compared to define the maximum efficiency that may differ by the system and the target species. So the suspended particulate matter can clog the filter like the organic matter or the precipitate and increase the concentrations of the PCR inhibitors. If the filter becomes clogged before samples are completely collected, researchers should record the sampled volume and optimize it for the use of new filters in the sampling. Carefully taken notes and sample records are essential to avoid mixing multiple filters independently of a sample. If clogging of filters is common, it may be necessary to use filters with larger pores for the study. Sampling strategy is very important. So if the sampling points are too far from the eDNA source, these species may not be detected. Therefore, the sampling strategy should be reviewed and the number of samples taken per station should be increased. As an alternative sampling strategy, sampling from different points around the study area is also something that is recommended. These samples are then mixed in a single sampling bucket for homogenization. A few samples are taken from this large volume of water to be transferred to tubes filled with ethanol and sodium acetate. Since eDNA is not homogeneously distributed in the environment, sampling from different points in the workstation, in the working sampling station, increases the chance of obtaining eDNA released from the target species. The most common strategy is based on filtering different water samples and is mostly applied in large and flowing aquatic systems. Since larger volumes of water can be sampled in eDNA studies using filtration, it theoretically increases the probability of detecting species. However, this does not mean the filtration always performs better than the sodium acetate method. The disadvantages of filtration technique are stated to be more time consuming and high cost because of the filters and the pumps. By filtering larger volumes of water, more eDNAs can be obtained. On the other hand, while the concentration of inhibitor in the samples also increase. So you must consider it also. The most important disadvantage is that since eDNA is mostly found in small fragments, The pore size of the filters may not be suitable for holding these fragments. These small pieces of eDNA may not be captured by the filters, thus reducing the probability of detecting species. There are 4 types of filters, mainly the most commonly used ones. They are the glass fiber filters, cellulase nitrate filters, carbonate filters, and the nylon filters, as I can remember. The sampling strategy should be adapted to the biology of the target species and the environment in which it lives. For example, a study stated that there are different detection possibilities between 2 different environments. You can see here, lakes and ponds and streams. They reported that the possible reasons for this difference is that the collection of 3 different water samples of 15 milliliters is suitable for eDNA detection in stagnant water environments, but this sample amounts is not sufficient in rivers, as you can see there. In flowing waters, where eDNA is more dilute, it is appropriate to use filtration methods that provide larger volumes of water sample. Figure shows the possibilities of detecting environmental DNA by qPCR in natural freshwater resources. So aquatic areas sampled and the percentage of identified species are given in the graph, as you can see. And it all changes according to the water type and according to the species that you are targeting. So the things to consider in terms of field. We should always use controls and negatives during the field sampling. This is important. And filtering or precipitation is a matter of choice, but filtering is mostly the most commonly used method in these days. Precipitation, because of its low capacity in terms of water that you can precipitate, filtering is more commonly used. The design should be made specific for the study, and the sampling strategy affects everything afterwards: the PCR, the high throughput sequencing, and bioinformatics. So without a good design in the field, your study is doomed. Decontamination is important. Using disposable gears or decontaminating them, sterilizing them in the field is important. Preservation is of the samples with cold or using some storage solutions is also important. The selection of pore size of the filters that you are using is another thing to consider. Inhibitors is something important. The more you sample, the more inhibitors you get into your sample. So you must always note the amount of the samples that you take. For the soil samples, the humic acid is the biggest inhibitor. For the water samples, again, there are lots of inhibitors in the environment. So don't forget that the more sampling doesn't always mean more results. It means more inhibitors, which will decrease the quality of your results. And you must always know the ecology of your target. So if you are dealing with, for example, a specific fish species, you must know where to find them. So looking them elsewhere in the environment will give you not good results. Let's continue with the laboratory applications. To obtain accurate and reproducible results, which is the scientific way, from eDNA samples, a strict decontamination protocol must be followed at all stages of the run, from analysis, reactions, filtration, to sample naming. So environmental DNA samples are susceptible to contamination like other low quality and quantity samples like ancient DNA and forensic DNA samples. As with studies of this type, eDNA studies should be kept and analyzed in a special room, or at least in a special cabinet, only used for that stage, and physically separated from the high DNA containing isolations and PCR reactions is recommended. All equipment and tools required to process eDNA samples should be kept in this clean laboratory environment in accordance with the contamination procedures, and items that are in common use, such as personal gears like laptops or pens, should not be left in this environment. The reagents used for the contamination of equipment and surfaces is critical to maintaining the cleanliness standards. For example, standard autoclaving is insufficient to destroy the nucleic acids. Among the contamination solutions, the sodium hypochlorite or the bleach is the most effective for removing DNA and PCR products. A 10% dilution of bleach, typically 6% sodium hypochlorite, is standard in many laboratories. However, the bleach diluted 50% is considered to be the most reliable value to remove unwanted contamination of DNA and the PCR products. Studies should not be performed on any surface that cannot be UV sterilized and is in direct contact with eDNA samples. After the eDNA laboratory is used, the work surfaces and the room where eDNA samples will be studied, like everything, refrigerator handles, centrifuge, pipettes, heat blocks, and surfaces should be wiped with 50% bleach and periodically cleaned before being used in the next study. Positive air pressure, air filtration, and UV sterilization, such as in the PCR cabinets with UV lamps and HEPA filters for the area of workbench, are the additional measures that can help to prevent the contamination. You can see the standard eDNA extraction steps in here: the lysis, binding, washing, eluting steps. These are mostly same for any kind of eDNA extraction. So for eDNA extraction, many studies in the literature use this blood and tissue kits. In some studies on eDNA extractions, comparison of these isolation or extraction techniques has been published. You can reach them easily. I will give you a list of references after this seminar series, so you can check them. They are really important studies that you can get really good ideas. According to those studies, it has been shown that these blood and tissue oriented kits are most of the time working better than water or soil specialized kits because of the inhibitor removal properties of the kits. But of course, it's all dependent on the optimization of the process, because most of those kits are using glass fiber filters in isolation without any target DNA. So it has been stated that more DNA can be extracted with the phase separation precipitation kind of DNA extraction methods, such as CTAP or phenylchloroform, than DNA extraction methods using silica columns, as in most of the commercially available kits. For studies requiring quantitative measurement of the eDNA, it should be noted that recovery of eDNA can vary between both filtration and extraction methods. So they should be optimized accordingly. However, the comparative performance of these methods under the effect of different inhibitors has not been investigated yet. So samples designed to be used should be stored at -20 or -80 in order to compare DNA extraction techniques that can be developed in the future. In terms of PCR and the quality and the quantity of PCR measurements, so the first eDNA protocols for detection of microorganisms were created by performing fragment analysis from conventional PCR products. More recently, prop-based quantitative PCR or qPCR methods have been developed and used for single species detection due to the sensitivity, specificity, and the quantitative measurement of eDNA in the sample. Recently, digital droplet PCR systems can be used when the purpose of the study is quantitative and may be a more cost-effective choice for very small and large number of samples if you are working in plates. When working with non-prop-based PCR systems like the conventional PCR or qPCR with cybergreen, and the possibility of cross-amplification leading to false positive results can be eliminated by analyzing the positive controls, and they should be confirmed by sequencing and together in the entire sample set. Therefore, the prop-based qPCR is currently indicated as the most suitable tool for eDNA detection of single or several target species. The use of high-throughput sequencing technique is considered a great advantage if the target organisms are not previously identified, so you don't have an opinion about biodiversity there and you would like to find it out. And if there is more than one target species in a study, again, high-throughput sequencing is the study that you should consider. The use of high-throughput sequencing in eDNA analysis obtained from environmental samples has a great potential for future studies on the monitoring and the design of the biodiversity worldwide. So in terms of PCR, you can see here that you can make tests with different polymerases because all have different inhibitor tolerance limits and all have different properties. Synthesizing rates. So from our studies, I can easily suggest that in terms of non-proofreading polymerases, you can use AmpliTac Gold polymerases based any kind of PCR mixtures. And in terms of proofreading polymerases, the Q5 polymerase can be used easily for environmental DNA studies. Quantitative PCR or qPCR is used to detect whether the DNA of species is present in the environmental sample with the species-specific primer and prop sets by targeting a small piece of DNA. It's typically about 50 or 150 base pairs long. Analysis performance for species-specific detection is based on the oligonucleotide hybridization property, which is a well-optimized but not precisely predictable structure. Sometimes DNA sequence databases such as GeneBank or BOLD are not sufficient for the design of species-specific analysis. Therefore, the qPCR analysis design may require the creation of local sequence databases. You go to the field, collect your own samples, Sanger sequence them, and build your own local database. Because in the databases, there are generally long sequences containing loci found in the mitochondrial genomes of many taxa. Studies should be designed and tested to be carried out in a clearly defined geographic area, thus limiting the species to which DNAs may be closely related to environmental samples. In order to increase the specificity of identification, analysis can be performed with primers designed for more than one loci for the target species. And the optimization of the design primers must go through 3 general steps: in silico, in vitro, and in situ, as you know. In the in silico test, primer performance designed by comparing DNA databases to detect possible non-specific oligonucleotide hybridization, and it is checked by computer-assisted analysis. multiple alignments of the DNA sequence of the target species with sequences belonging to closely related and unrelated species are recommended using these designing programs. For in silico validation, bases on each primer and probe are required to prevent cross-amplification when target species are rare or absent in the DNA. In vitro tests are the testing of primers or probes designed on DNA obtained from the tissue of the target species. So first we designed them in silico in the digital environment. Then we test them using a really reliable DNA extracted from the tissue. That's the in vitro case. So cross-contamination between the DNA extracts from tissue is easily encountered in vitro tests. And at the last stage, in situ testing, it requires verification of primers or probe performances on the real samples, like from eDNAs extracted from filters or environmental samples. One of the biggest issues when you are working with environmental samples is the inhibition. Inhibition encountered in PCR reactions usually occurs frequently in eDNA samples and results in unsuccessful or delayed amplification of the DNA of the target species. If you're using high throughput sequencing, this process can be observed very clearly during the library preparation phase. If you're using the qPCR analysis, this situation appears as the absence or the shortage of the target species. In order to detect the PCR inhibition, the positive control DNAs on DNAs of another species known to originate together with the sample analyzed during the reaction must be amplified in the PCR device. These can be classified as positive controls, or you can use spiked samples, which means you can include some positive control DNAs in your eDNA samples to see if your reaction is inhibited by the environmental DNA or the inhibitors in the environmental DNA, or there is something wrong with the design of the study. These are known as control reactions, internal positive controls, the IPCs, or internal amplification controls, IACs. For eDNA samples, using a low amount of IPC, internal positive control, matching the expected eDNA concentrations may best reflect the degree of inhibition of a reaction, as it will not amplify at least approximately the same level of inhibition. Alternatively, when samples are analyzed in triplicates in qPCR, the inter-repeat CQ values change is accepted as evidence of inhibition. You can see from the CQ values here, if they are, if you dilute your samples, it will give different CQ values in terms of amplification. So the more DNA you have in your sample, the amplification starts earlier. The less DNA, it will start amplification later. But if inhibition is there, then more sample means more inhibitors. So it will start to amplify later, or it will never start to amplify. So you can analyze it. Before analysis, these internal positive controls should be tested using the multiplex analysis. In addition to all these, it has been shown in studies that dilution of DNA reduces the inhibition rate during amplification. So another option is PCR inhibitor removal kits. They can be also used to solve this problem, but may also result in the loss of target DNA. So alternatively, various modifications can be made that can be applied to PCR reaction protocols, such as the use of bovine serum albumin, BSA, and inhibitor-resistant polymerases. As you can remember, the minimum information values for a metabarcoding study, there are also some criteria or some processes that you need to verify your analysis if you are using the eDNA positive term. So in order to accept it scientifically eDNA positive, there are some levels of assays or levels of protocols that you need to check with your design of the study. There is currently no established criterion for the minimum proportion of positive eDNA samples or a constant for the number of samples that must be taken to reveal the presence of species. But the detection power of eDNA depends on the frequency and consistency of positive eDNA samples taken from somewhere, together with information about the distribution, habitat, and behavior of the species, and information about the ecological, hydrodynamic, and other important characteristics of the system. For example, a single positive sample provides weak evidence of the species presence at the designated location compared to multiple positive samples collected over many years. However, when the initial concentration of target DNA is extremely low, like below 100 copies per reaction, it also creates discrepancies between qPCR copies. The number of PCR repeats, the technical repeats in eDNA studies varies from 1 to 12 according to the studies. If only one well is positive, it should be remembered that these results should be interpreted carefully and accordingly. So, one of the most important cases is the primer selection. There are many new studies focusing on the extraction, preservation, and the isolation of DNA. When these studies are examined, it's revealed that many of them talk about the issues that researchers should pay attention to and test before conducting a study for a healthy DNA progress. The primers used in the PCR step of molecular-based species identification studies on animals and plants or microorganisms often target mitochondrial, plastid, or nuclear ribosomal RNA genes. The standard barcode regions defined by the Consortium of Barcode of Life are cytochrome C oxidase subunit 1, or as we know it, CO1 or COI, for taxonomic classification of animals, and ITS for plants, or 16S for microorganisms, again ITS for fungi, or for plants, ITS is used with the combinations of RBCL and MATCAI loci. However, there are some limitations in the use of standard barcoding regions for eDNA metabarcoding studies designed for macroorganisms. There are difficulties in identifying with a single universal primer specific for the CO1 gene region, with the discrimination power of all the taxa. Although researchers conducted studies to design different primers for different taxa, they could not find the suitable primers due to the length of the gene region and the decrease in the power of discrimination between species. Therefore, the primers belonging to different taxa from different gene regions are used in eDNA studies. The most common alternative barcode regions used are the mitochondrial ribosomal genes, such as 12S and 16S, or protein coding genes, such as cytochrome B. Specifically for plant barcode loci, the combination of the two loci, like RBCL and MATCAI, are used for DNA barcoding studies from tissue in particular, and they are amplified independently. However, it's not always possible to find out which fragment the genes are associated with in the DNA sample, which makes it difficult to identify the species using the standard plant barcode in eDNA studies. Bioinformatics can help solve these situations to some extent, and can be useful when species diversity is low in the sample. Therefore, one or more barcodes are often used, like the P6 region of the tRNA intron. In addition, some loci have the high evolution rate and do not encode protein and can be used, such as the ITS gene region in the ribosomal RNA. So, however, because these barcode regions have intragenomic variation that makes their use in biodiversity studies difficult, they do not always allow alignments to be made to identify OTUs during data analysis. These loci in sequences of unknown environmental origin are often excluded from analysis unless there is an exact database matched down to know and validated biodiversity information. Due to these factors, in studies requiring phylogenetic analysis, other metabarcoding regions such as 18S rRNA genes may be more suitable, especially to narrow taxonomic definitions and overcome the database limitations. After selecting the locus or the loci to test the research-specific hypothesis, the primers in the study are designed universally or specifically according to the relevant taxonomic groups. When selecting pre-designed primers or designing new primers, rigorous in silico, in vitro, and in situ testing is the most important to reveal the advantages of a new eDNA metabarcoding study. Another important factor is the size of the amplicon to be replicated. Since the applicant size is one of the primary steps in the success of the species identification, it can affect the result of the study one-to-one directly. So, however, since short DNA fragments are more likely to be found in the environmental samples, the amount of DNA that can be obtained will be higher than long fragments. In addition, the use of more than one loci for the target groups allows for consistency tests between loci and increases the likelihood of species identification. Sequencing multiple samples on the next generation sequencing platform after the primers are designed for eDNA metabarcoding work and desired gene regions are amplified during the PCR stage, it provides a significant advantages in terms of costs. Nowadays, next generation sequencing platforms are preferred more than other next generation Sanger sequencing platforms due to their error rates and other stuff. So Illumina platforms are preferred than the platforms such as IonTorrent or Nanopore because it has lower error rates. Sanger sequencing has the lowest error rates, but they're completely different in terms of metabarcoding. So one of them can be available for barcoding, the other is for metabarcoding. So in the laboratory checklist, the experimental design must include enough biological replicates, enough technical replicates, and many extraction controls to detect the potential sporadic contaminations, many negative PCR controls to detect potential sporadic contaminations, and many controls to estimate the importance of tag jumps, and many controls to estimate the importance of index jumps in terms of high throughput sequencing. The probability of detecting the target animal organism is important. The suitability of the protocol applied to the detection of the target species or livings must be tested from sampling to analysis. Testing for this probability is crucial to the interpretation of possible false negatives. You cannot be sure is there really a target species or a method is not suitable. Another issue for the checklist is the number of technical repeats created in the PCR. The number of PCR technical repeats, technical replicates performed on all samples affects the probability of detection of the species. So it's recommended to use a larger number of repeats, like 3 to 12 PCR repeats during the amplification stage to increase the probability of detecting the target organism is important. Another issue is the inhibition control. We already discussed it. The organic materials in environment can bind to DNA and hence it can inhibit the PCR amplification. Isolation and PCR of samples taken for negative on positive controls is important. So when you're designing your plate, when you're designing your study, including blank samples, positive controls and negative controls takes an important place. Ecological knowledge is one of the other things that you need to consider before you go to the field, because obtaining information about the ecology and behavior of a species is very important in designing the sampling strategy, where and when to sample. Another issue is the suitable laboratory design. So for eDNA studies, a number of serious precautions should be taken to prevent contamination, as in laboratories working on ancient DNA or forensic DNA. So many steps should be separated from each other: the field, DNA extraction, PCR, and sequencing. If you can't find different rooms only dedicated to these stages, you can use dedicated cabinets to use. qPCR is another issue that, since it works more precisely and more specifically, qPCR is preferred over traditional PCR. So using only a target species specific probe rather than the specificity of just the two primers will drastically reduce the amplification rate of the false positive results. Multispecific approach is consisting two methods: species-specific approach, as we discussed, like in the qPCR, and the universal approach, as we discussed with the high-throughput sequencing approach. And the last issue is the reference databases. A reliable reference database should be used to compare the sequencing data. The general databases that we know, the NCBI, GeneBank, EMBL, Bolt, Silva, are useful for generating preliminary information, but often include sequences entered with incorrect descriptions or incorrectly sequenced data. So local reference databases, on the other hand, provide a more reliable source, since they are created after verification and approval, of course, if you create it this way. So depending on the research question, there are different ways to follow it. So I leave this as an example workflow showing 3 different environmental DNA research questions and experimental procedures that you can follow. So before we move to bioinformatics part, I will give a 10 minutes break and we will meet at 12:15 for a short final lecture on the bioinformatic parts. If you have questions during this process, you can write it from the chat or however you wish. So I'll be here, just closing my camera now, and I'll see you at 12:15.